to everybody. It's really a pleasure to be here, and I'd like to thank you, Marcia and Professor Steve for the invitation. And I'm going to show you a part of our work that we have been developed at the medical faculty of the Universidade of São Paulo here in Brazil. So we are these last 6 years focusing one very dramatic neurodegenerative disease. It's called amyotrophic lateral sclerosis. Amyotrophic lateral sclerosis is the third more prevalent neurodegenerative disease in the world. The disorder is related to a neurodegeneration of both superior and inferior motor neurons. The disease can compromise patients from 20 to 80 years old. The diagnosis of the disease is very difficult. There's no biomarker, and the disease present a very severe disability and high morbidity. So it's really a dramatic disease, and there's no specific treatment. Only one drug approved and used around the world that is called Hiluzol, is a glutamatergic antagonist that is said to be it's considered that increase the lifespan of the patients for something like 2 or 3 months. So there are two clinical forms of the disease: the sporadic one and familial forms. Sporadic form represents 95% of all ALS cases, and familial one about 5%. The familial forms of ALS is related to the mutation of several different genes. Until 4 or 5 years ago, the most important mutation was related to the superoxidase dismutase 1 gene. And all knowledge that we have regarding amyotrophic lateral sclerosis are related to the studies in this animal model. A model that represents something almost minimal regarding all clinical features of amyotrophic lateral sclerosis. So it's really dramatic. Something has to be done, and start to be. For example, some organizations have evaluated all experiments that has been done in most important drugs that have been done in animal models, and you can see that the new results is really different than the previous results that supported the application of several drugs, including the most important one, Hiluzol. Some of them, like for example lithium, that it was mentioned, positive results around 15%. Now they have been describing negative results employing this compound using animal model. So when you have these patients, their families, in that dramatic situation, all the time they ask, "Well, but you are the research, you the team, you are the research. You are not doing anything that can really change this dramatic situation." So it's a environmental pressure that we receive all the time. So there are several unmet needs for ALS. The most important one that is considered nowadays is the need to discovery of a better clinical model to study the disease. So all laboratory discoveries, the hope of new discoveries in the disease is now on the development of new preclinical models. Okay. So, to try to do something, we have developed a system to investigate ALS in our unity, in our hospital, that we call it ALS Translational Neurology Program. We have started with the assistance at the hospital and set up several laboratory units to develop, to perform systematic and connected study to try first to understand the mechanism related to disease and try to develop new clinical targets for it. So, as the responsibility to get results in terms of knowledge on neurodegenerative mechanism and new clinical therapeutic targets, we have used samples from ALS patients. All studies approved in several regulatory agencies in our country. So, here I'm going to show only two aspects of several experiments and analyses we have done in ALS program. Taking into account the knowledge or hypothesis on dying back events in the disease related to signals that may appears in the muscles or in the axon terminals that could be related to neurodegenerative process, we have decided to evaluate it in a very detailed way the human Schwann cells obtained from the motor nerve that's going to be, destroyed by the progression of the disorder. So, we have developed the technique to purify Schwann cells in several methodologies I'm going to tell you, and perform molecular analysis in these cells, in these human cells, and also to take the neighbor cells located close to the Schwann cells, that is the fibroblast, and fibroblast in the nerve plays a role in the trophic maintenance of Schwann cells that are responsible to maintain the trophic state of the motor neurons. So we have used fibroblasts of the motor nerve in a phase that the motor nerve was still functioning to transform them and inducible pluripotent stem cells and then motor neurons for our studies. So it's the first time that has been done in the world in a trial to do not use the familial forms that in fact don't represent the majority of the case. So we have obtained enriched Schwann cells by laser microdissection, cell cultures or flow cytometry. We have developed this methodology to obtain Schwann cells from human nerve. Demonstrating the enrichment by molecular analysis, and then the analysis of gene profiling, specifically in the Schwann cells. So, analyzing the gene profiling, the RNA expression, we have evaluated molecular aspects, biological aspects, downstream aspects, and the analysis of the overview workflow data, and depicting some pathways involved and characterized new methods. Perhaps I will have time to say something about that, and evaluation of new therapeutic targets. So, just one example of the large amount of results that the methodology can bring to you. Here we have demonstrated that Schwann cells, in terms of growth factors, many of them are play actions on neurons, or we can call them neurotrophic factors, are deregulated. Many of them are downregulated, and some of them are upregulated. So it's very interesting that we now know that some neurotrophic factors expressed by Schwann cells are downregulated in the human disease. We can also analyze other possibility, other signaling pathways. For example, cell adhesion and focal cell and focal adhesion molecules, that is the pathways that are extremely compromised in the disorder, in the human disorder. And the molecules have been evaluated. You can combine both pathways and the involvement of the molecules in those deregulated pathways, and depicted the molecules, for example, that are common to both pathways, and you can start evaluating the importance of those specific molecules in the neurodegenerative process. Some hypotheses have been constructed by our groups regarding these results. Okay, but how can you test that? How can you test the influence or the importance of those molecules, of those deregulated pathways? You can now use the advantage of the transformed human motor neurons to test all these hypotheses. So we have done that. As we were interested in translational aspect of the disorder, we didn't go, as I said, by the classical model using mutant cells or cells carrying human genes, human mutant genes, etc. We have used cells from patients of the sporadic forms of the disease. So, from the biopsy of the nerve, of the motor nerve, here in the foot, we have obtained fibroblasts, and those fibroblasts were adult fibroblasts, were reprogrammed. We used a control by patients. I can explain this for those that are interested. And those cells were reprogrammed in induced pluriprotein stem cells using two different methods. We have used the classical one, the cytotulus sendivirus vectors from Life Technology, and one, a kind of new one, that is human Stenka lock-speed lentivirus. This is the first, there was, not now more any longer, but there was the first time in the world that someone had obtained an iPS from sporadic patients. That was published 2.5 years ago. So, those iPS were afterwards submitted to a specific program to be transformed in motor neurons. And those differentiated cells were done for the first time in our country. That's the demographic table of the patients. And we have several patients that we could perform a quantitative analysis. Interesting, we have done that in collaboration with the Professor Yamanaka. Some of my students spent some time in Japan, but and there it worked very, very nice. But here in Brazil, we reprogrammed the cells, but we could not get the large amount of cells that we needed for quantitative molecular evaluation. So that's the reason we have changed the system for STEMCA 1. STEMCA is a crescitable constitutive polysomic retrovirus expressing the embryonic genes. So here are the results, the marker of pluripotency here as well. And very importantly, to do quantitative experiment, quantitative analysis of the pluripotency, you'll find very interesting results here. And here the spontaneous differentiation of the cells in the three germ layers, both in vitro and in vivo, in the classical way that should be done. And then, after demonstrating the pluripotent capacity of these cells, they were applied in the specific program to be transformed in motor neurons. So we have done that. It's very interesting, the effectiveness of the methodology. We have obtained quite a lot of motor neurons. It was very surprising, actually. And those motor neurons were evaluated rigorously in terms of gene expression of embryonic markers, adult markers, and so on. Control experiments to observe eventually a specific presence of cells there. And very importantly, the vital state of the motor neurons. Here, only for fun, some iPS were developed in cardiomyocytes, working cells, but we were not interested on that cells. So, those motor neurons were evaluated again using large platform of microarray with 6,000 sequences, and in a classical way, as you know, and were presented here. And in this analysis, we have evaluated the deregulated genes, those that show upregulation of the mRNA expression, and those that show the downregulation of the mRNA expression. And that's it. The genes were submitted to biostatistical analysis, and in here I am talking about the gene expression of motor neurons, comparing motor neurons of the ALS patients and the control. In this experiment, we have evaluated more than 1,000 deregulated genes, 1,000 downregulated, and about 500 upregulated ones. And then we went to evaluate the functional aspects of those deregulated genes. Evaluating the classical way, the KEG pathways, and the gene ontology terms that show us involvement of biological process, molecular functions, and cellular contents. And very interesting, we have observed that those deregulated genes show two pathways in the KEG, system. The Parkinson's disease and ALS sometimes. ALS patients sometimes present signs of Parkinson's disease, and importantly, oxidative phosphorylation. In terms of gene ontology, gene ontology analysis, looking at the biological process, molecular function, cellular components. All these analyses together show high involvement of mitochondrial-related genes in the motor neurons of sporadic ALS patients. So the genes were described in the paper. We have validated several of the regulated genes and try to describe other functions. For example, RNA processing, RNA metabolism, and intracellular transport, and so on. And in all these subtypes of cellular components or cellular function, we have found the regulated genes related to mitochondria or mitochondrial function. So it's very interesting. We are not postulating that ALS is a mitochondrial disease, but mitochondrial dysfunction is very important, seems to be very important to the neurodegenerative process. And these mechanisms were not described in animal models so far. And now we have evaluated the network of those deregulated genes in terms of functional protein association. Is very interesting. We can look at the correlation of some proteins related to other protein, and this could describe or show us some important silent pathway deregulated or altered, involved in this system. The same for physical interaction analysis in the network using specific tools for that. And the genes depicted are there in terms of high degree of importance in the network and high betweenness in the network. And all these informations again are submitted to new statistical bioinformatic analysis. Co-expression is a little bit complex, but we can go on to try to depict some highly important mechanism or involvement here in this complex network. And the genes and the molecules involved are there. So, after all this knowledge, we have combined again the analysis of the gene profiling in the human children cells of the disease and the transformed motor neurons in the disease. And interestingly, we have some specific algorithms that reinforce the individual patient data in the defined context. That's my, we have only one slide more, I'm almost finishing. So, now we are able to select some molecules, and we are right now evaluating the effect or the importance or the impact of those selected pathways and molecules in terms of neuroprotections in an in vitro model. So here, for example, we have motor neurons in the absence of a specific factor. And here, the motor neurons in the presence of a specific factor. In the absence of a specific factor, you see, a large amount of apoptotic signal in this medium. And we don't see those apoptotic signal, or we see very little apoptotic signal in this system. So it's very interesting. And more interesting is that when you observe that these responses are quite substantial related to specific patients and to a specific clinical feature of the patients. So with this data, we imagine that we can in the future develop translational neurology based on personalized medicine. As many of these molecules are large molecules. Large molecules, sometimes you have problem with systemic administration, you know, the blood-brain barrier and so on, and side effects. So, as we are conducting this, the first clinical trial of mesenchymal stem cells in the country, phase 1, phase 2, and mesenchymal stem cells are very well known to be able to synthesize and to secrete specific molecules that interact with the inflammatory medium, that promote neuroprotection and so on, modulate triggers immunomodulation events in the system. We use that features of Schwann cells to try to stimulate them in vitro and see if the cells, the mesenchymal stem cells that are collected from the bone marrow, if those cells are able, after specific stimulus, to secrete specific molecules for motor neurons. We have obtained some high expression of some neurotrophic factors, some not neurotrophic factors that are neurotrophic factors to motor neurons, but we are now obtaining results with neurotrophic factors that are not exactly specific of the best neurotrophic factors for motor neurons. So those secretion, the secretion, the mesenchymal stem cell secretion, are tested on transformed motor neurons. So, as we saw in the last slide, we are now obtaining some aspects of neuroprotection in the transformed motor neurons. So, we think that if this history goes to the direct way, we can, in the future, we are maybe be able to test these new molecules using this system in clinical trials. Thank you very much. To visit the Universidade of São Paulo, in São Paulo, and the medical faculty that is here, the Universidade of São Paulo, in the campus, in a specific place in São Paulo, and here the clinic hospital is the biggest public hospital in Latin America. Thank you very much.